In the insect ecology group, same as Rosa earlier today, but I work on museum collections and looking at long-term change. So I suppose it's kind of like honed in so at this point that globally we're having incredible impact across all the natural environments around. And we're kind of able to understand that how biodiversity is responding. And I thought I'd just kind of talk about how we actually know that. Well, we know that because we go through these sort of biodiversity monitoring surveys. So Rosa talks about some of those as well earlier on. So in the UK, we have some of our longest running surveys, so be that for birds. This is run by British Trust for Ornithology. Lots of volunteers all across the UK spend a lot of time bringing birds. And here there's a blue tit as part of the constant effort scheme. That's gone back to 1983. That's where the first ones start. And then we have butterfly monitoring as well. The UK butterfly monitoring scheme goes back to 1976. It's probably one of the best in the world. Here we are not doing a butterfly transect as part of the butterfly monitoring. This is just my research group. We were catching some butterflies, doing some sort of, well, we were doing the kind of the work that Rosa was talking about, trying to catch butterflies and measure their temperatures. But I thought that was a nice picture anyway. So from these sort of biodiversity monitoring schemes, we're able to essentially assess how biodiversity is responding to the changes that we are making to the natural world around us. But once these schemes sort of start about 40 to 50 years ago, we know that we've actually been altering the natural environment for far longer periods of time. So in fact, land use changes occurred for centuries and things like that. And we also know that not butterflies, biodiversity also changes a lot and is able to respond to changes that we make to the natural world. And roughly speaking, this is kind of a dummy diagram I've drawn, but this is kind of what long-term biodiversity change kind of looks like. And it matters essentially where we pick to measure our trends from. So if, say, this is 20 years worth of data, we might get very steep declines. If we look sort of 40 years in the past, we might get a sort of slight increase. And then if we just randomly pick bits and pieces and going further back, we get very, very different severity and direction of changes. So having a long-term perspective allows us to contextualize the recent changes that we're seeing and to understand how severe these are, how long have they been going on for. And this is kind of roughly where sort of my sort of interest goes. So I suppose it's not a surprise I work in museum collections, and they're really quite fantastic sort of tools to understand how we can extend these time series. So museum collections roughly begin sort of in the 16th century, sort of as cabinets of curiosity, so people just collecting lots of things. And then in sort of the 18th and 19th century, you get it's very closely tied with advancements in taxonomy, botany, and zoology, and you get really huge collections being built. And then today there are over 1,000 museum collections, including that, of course, in this museum, holding between sort of 1.6 and 2.1 billion specimens. I expect it's actually probably more than that. That's just a recent estimate. So these are some of the butterflies that I've been really lucky to work on. And so what do museum collections actually give? So I work on insects. I suppose you won't be surprised to know that. But basically with collections we're able to have lots of fully preserved individuals, and these essentially allow us to understand a little bit about the genetic variation, the morphological variation within a given species. But often, excitingly, they are accompanied with specimen labels, which give us an idea about where and when a species was collected, in addition to some other exciting information as well. And by having this information about where and when species were collected, then we can begin to kind of track changes across time and understand how species and communities are changing over time. Sometimes we also get cool things like, so on the left you see a coral that's from Thailand, held at the Natural History Museum, and that red line that you kind of see in the cross section, that's from an iron pollution event that occurred in a nearby factory. So that gives us a picture of the environmental conditions that were around the specimen at the time. And then here, this is one of my favourite specimens in the museum here. You can also get things like pollen loads, so you can understand the interactions that species were having with their contemporaries at the given time. And so for the next final bit of this talk, I thought I'd give sort of a overview of some of the work I've done. I'm not going to go into the methods. I'm just going to give you explain why I've done, the context behind it, and some of the key findings. So hopefully that's somewhat interesting. So the first piece of work I did in this sort of area, which really got me excited into working on museum collections, was done here with my colleagues Matt, Ed and Tom, who's now at the NHM but was here back then. So we are currently in that dark blue dot in the middle. And historically we would have been a huge expanse of wetland habitat, so the fens all around us, so it's this huge wetland habitat. But around 1850s, we had sort of 99% of the fens were drained. This was mostly associated with the advancements in steam engines and making it actually possible to drain it permanently. And this land obviously around us, I suppose you won't be surprised, is that there is all this agricultural land because it's incredibly fertile, because it used to be inundated like constantly. And some of you will be aware that we've got really exciting projects all across the area to try to restore some of these wetland habitats. So we've got the Great Fen Project, which is really exciting, and you can read about it all online. But one of the issues that we have associated with these sort of restoration projects is we don't really have benchmarks to measure success against. So we don't have biodiversity monitoring surveys going all the way back to 1850s. And so it's kind of difficult to do that, assess those. And this is kind of what our work kind of goes into. So we focused in on aquatic insects because aquatic insects have both a juvenile aquatic stage and a terrestrial adult stage. So the drainage of the fens quite likely had a very large impact on these insect communities. And so I'm going to go through two of the main findings. So one of them was that we saw that in Cambridgeshire, so we focused on Cambridgeshire aquatic insects, and we found like these communities were restructuring. So we lost these lentic specialists and had lots of lotic specialists. Just go to explain a bit what these terms mean. So lentic is roughly sort of stagnant water, so the thing that lies sort of standing water, and lotic is sort of flowing water. So it's essentially with the drainage of the fens, it's not surprising that we lose all these specialists that are specialists on sort of more stagnant standing water that you see in wetlands, and moves more to sort of the lotic specialists that you perhaps see in sort of drainage channels between sort of fields and things like that. And then the other cool thing that we found was, so we only looked at this for the diving beetles because we didn't have enough amounts of data to do this with the other groups. But for the diving beetles, what we found is that the restructuring that we saw in the aquatic, in the community, in this sort of switch from the lentic to lotic specialist, that didn't occur in Wicken Fen. And so Wicken Fen is a nature reserve. It's the only area of the fens that wasn't drained. And so lo and behold, it's not surprising, I guess, or actually an incredibly good news story is that we don't see those changes seen in this area. So moving on, so I was very interested in museum collections after this point, and I went off to do a PhD up in York, but on a group of butterflies that was very far away from here instead. I was looking at how museum collections could help us understand tropical biodiversity change. So these are all endemic species from the island that I will show you in a second. But for the areas of the world where biodiversity is richest and where we have the fastest rates of habitat change currently, we know incredibly little about how tropical biodiversity is changing. And this is largely because we don't have very many data sets, and resources typically have been much lower in the tropics. And these monitoring surveys that we have in the tropics now have only just been started. So this postdates most of really large-scale environmental changes that have occurred in these regions. And so my work is looking at how we can use museum collections to help this. And this island is Sulawesi in Indonesia. It's an island of exceptional endemism, this meaning that there's a very high proportion of these species that occur on this island that occur nowhere else in the world. And that's the same for these three species, the black-crested macaque, greenback kingfisher, and the flying dragon. And of course, there are butterflies. 40% of the butterflies on Sulawesi are endemic. These are my favorite butterflies, Papilio blumei. I mean, they're just beautiful. I mean, who could say no? Just to give some sort of insights about what I found during my PhD. So what we actually found was that because we were working on this island that had really high endemism, we had some really quite strange things. So endemic species are typically seen as more vulnerable to climate and land use change, but actually on Sulawesi we have these endemic species filling sort of different niches because we have a lot less of these wider spread species. So we find that actually it's sort of them being montane, them being actually habitat specialists that actually make them more vulnerable rather than endemism per se. We were able to create 164 years of a time series using museum collections, which is really, really far back. And basically what we see is we don't see systematic declines in species richness, but what we see indeed is that we see increases in host plant generalists, we see increases in species that are tolerant of open habitats, which is consistent with the fact that they are more tolerant of human modified landscapes. In the present day. So it's a much more complex picture than often that's dramatized in the media. We also found that when we account for historical records, we can see much larger thermal ranges that species occur at. And so actually, sometimes species actually occur at hotter temperatures in the past than we currently see them today. And that kind of, it's kind of slightly, provides some level of optimism in that actually they might be more resilient to some of the warming that we have, but it requires sort of, well, it's more nuanced than that, but we might have to recreate some lowland forests in order for them to actually occupy those habitats. And finally, just to quickly move on to my current project, which I'm based here at the museum. So we are living in an increasingly lit up world. So light pollution is increasing about 7 to 10% each year. But what we know that light pollution has a large impact on the natural light patterns that many biological processes depend on, but we don't really know about the long-term impact it has on species, communities, and wider ecosystem services. And so that's kind of what I'm working on at the moment. I've just started, and basically we have collections that predate the advent of street lighting, and then we have various changes, transitions throughout the sort of 2 centuries. We have the blackouts of the Second World War. Do communities go back to how they were before we had lights, or do they transition to something completely different? We don't really know that now, and I'm hoping to find that out pretty soon. Yeah, so thank you.